Welcome to the Faculty Focus podcast, live from our annual Faculty Research Day with your hosts
Marleen
and
Teun,
highlighting the research of our professors and PhD researchers.
Hi again. Our next guest is Assistant Professor Michaela Prothiwa, specialized in chemical biology at the Group of Medicinal Chemistry at the University of Antwerp. Her research focuses on applying chemical biology to understand immune-pathogen interactions. Welcome to our podcast, Professor. Your topic of today is a chemical lens on infection and how probes help us understand bacterial virulence. Can you start with explaining the concept of bacterial virulence to a non-scientist like me?
Yes. Yeah, thanks for the invitation. So bacterial virulence is actually the ability of bacteria to make us sick. You know that in our body there are a lot of friendly and good bacteria, but there are also bad bacteria. And we have to know actually why do some bacteria make us sick and others not. And actually the virulence is the ability of the bad guys to make us sick. So that's the difference between the good and the bad ones. Exactly.
Okay, That was clear. Thank you.
Well, and for me, it feels like it's very new, actually, targeting the bacterial virulence instead of the bacteria itself. Is it new, or is it just me being out of research for quite a while?
No, actually, it's quite new. So,
Okay.
as you know, traditionally, we have actually relied on antibiotics to treat infectious diseases. And we have used too many of the antibiotics, and this has led to a big problem, the antibiotic resistance crisis.
Yeah.
So actually, meanwhile, we have a lot of antibiotic-resistant infections, and we really need somehow new strategies to solve this problem.
Yeah.
So it's really a problem. And we need alternative strategies, and one of them is targeting the virulence.
The virulence itself. And it's also consisted about or around different parts, the bacterial virulence. Is there a specific part that you focus on?
Yes, so actually bacteria produce a lot of secret weapon or specialized tools, so called virulence factors. And they have actually a lot, but some of them are especially important. And one of them are actually proteases, so that are proteins or enzymes that break down our body's proteins. And bacteria use them a lot to, for example, degrading our tissues or to break down our body's immune responses.
Nasty things.
Yes.
So your research focuses also on the activity-based probes. How would you explain these things, the activity-based probes, to your grandmother? Is there a simple everyday comparison?
Yeah, it's a very chemical strategy. And it's very tricky to explain this because we use chemistry to understand biology.
All
So
right.
actually these chemicals, so that's molecules, you can see them as a chemical lens that zooms into the infection process. And these chemical lenses will show us when are these bacteria using their virulence factors and when not. And only when we understand how the bacteria make us sick, so when do they use the virulence factors, only then we can develop treatments against them.
Yeah, and intervene at the right time, at the right
Exactly,
place.
yeah.
Yeah, all right. So I've seen in your abstract as well that your research covers a lot, from understanding bacterial pathogenesis to finding new drug targets for antibiotic-resistant strains. So do you think that the concept of bacterial virulence will replace the use of antibiotics?
I think that both of them have to go in hand with each other. So we should not focus on one of them. I think it's important to have antibiotics, and I think that's one of the most important drugs we have. So we cannot get rid of that. But we can have alternatives that maybe, to basically as an alternative strategy. Yeah.
Yeah, all right.
To use the two strategies together, or
For example,
okay.
Or to use in certain cases one of these strategies. So, for example, you could use antivirulence strategies to detect the bacteria and then use the antibiotics more efficiently or not too much or in the right way, for example. And antibiotics, if someone is really sick and nearly dying, you cannot, then you need an antibiotic, you have to kill the bacteria, you cannot wait.
Of course. Oh, true.
Yeah.
Right. Do you actually feel that we've all become more germophobic since we've been introduced to COVID?
Actually, I think in one way it's a good way that COVID happened, because people are more aware now that we can have pandemics. Yeah? So we had also pestis, or we had bacterial pandemics too. And if now the antibiotics do not work anymore, we have a post-antibiotic crisis, meaning, what will we do? We will all die from simple bacterial infection, and this is very dangerous. Yeah.
True. Yeah. Well, there is actually another thing, a side note. We've seen the graphics of your presentation. They look wonderful. How important is that aspect for you, the graphical side?
I find it very important because pictures explain a bit better what's happening. And I'm actually also active in science communication, and I like to create these graphics myself to explain this.
So you did those yourself as well? Yes. I think they're wonderful. I wouldn't want to be one of the knights on it. Anyway, I think they won't survive. Before we wrap up, one last question. What advice would you give to young scientists at the beginning of their academic career?
A very important fact in science is actually that you have to go out of your comfort zone. You have to try new things.
So a bit like we're doing now today with podcasts.
Yes, exactly. Okay. Yeah, because only when you go out of your zone, there is actually where the things happening out of your comfort zone. So
that could be a good tip for our listeners as well. All right.
Yes.
Thank you very much for coming, and good luck with your presentation this afternoon.
Thank you.
Bye.